সুলক্ষণা ম্যাডাম শুরু করতে পারেন। আমি তো দিয়ে দেখি, মানে এখন তো সতেরোপতি কথা বলতে পারবেন।
সুলক্ষণা দি, unmute yourself.
সুলক্ষণা তুমি আনমিউট করনি। সুলক্ষণা শোনা যাচ্ছে না। সুলক্ষণা ইউ আনমিউট ইয়োরসেলফ। এই রে, সুলক্ষণার কথা শোনা যাচ্ছে না। সুলক্ষণা আনমিউট ইয়োরসেলফ। এই শুনতে পাচ্ছ কেউ সুলক্ষণাকে?
না দিদি, না দিদি, আমি কল করছি, শুনতে
শোনা যাচ্ছে না।
She is unmuted. Somehow she is having an audio problem right now.
আনমিউট তো করা আছে কিন্তু অডিও আসছে
না। না, আমি তো দেখতে পাচ্ছি ওর মিউট করা।
হ্যাঁ, আমিও তো দেখতে পাচ্ছি ওকে মিউট করা।
কিন্তু সুদেশ না, দেখো এখানে আনমিউট দেখাচ্ছে, মানে তোমার এই যে পার্টিসিপেন্টস লিস্ট, হ্যাঁ ওইখানে আবার আনমিউট দেখাচ্ছে। আর
না, কিন্তু এখানে স্ক্রিনেতে মিউটেড আছে।
হ্যাঁ,
ঠিক আছে অমিতা দি, আপনি বলুন, আমার মনে হয় শেষ হয়ে গেছে।
কিন্তু ও মনে হয় তাহলে কারুর কথা শুনতেও পাচ্ছে না, কারণ আমরা যে বলছি এত যে ওকে শোনা যাচ্ছে না, সেটাও তো শুনতে পাচ্ছে না।
আমি দেখতে পাচ্ছি ও আনমিউট আছে, আমি যতটা দেখতে পেলাম।
কিন্তু এখন তো আমি দেখতে পাচ্ছি এখনো আনমিউটেড, কিন্তু আমিও কিছু
এই যে ও লিখছে, ওকে নো লিখতে হবে।
লিখেছো নো?
হ্যাঁ, শ্রীতমা লিখেছে।
অমিতা দি, আপনি বলুন, মানে অন করে। আপনি কিন্তু
সুলক্ষণা ফোন করেছিল, আমি ওকে বললাম একবার মিটিংটা লিভ করে আরেকবার জয়েন করতে, তাহলে মনে হয় ঠিক হবে। ওর অডিও প্রবলেম হচ্ছে, ও আমাদের কথা শুনতে পাচ্ছে না অ্যাকচুয়ালি।
Hello, is it audible now?
Yeah.
Yes,
perfectly.
এখন শোনা যাচ্ছে?
Yes, you are audible. একদম শোনা যাচ্ছে এখন।
Okay. Sorry for the interruption. Let me start it again. Good morning, a very warm welcome to all of you to this program. We have organized a Student Week celebration as part of the initiative taken by Higher Education Department, Government of West Bengal. This is the second day of this celebration, and we have with us eminent scientist Dr. Shomiran Panda, Additional Director General ICMR. He will be delivering a webinar on awareness about present pandemic with special emphasis on Omicron. So now, without wasting any more time, I would like to welcome Dr. Amita Kaur, TCS, for delivering the welcome address. Amita di, please.
Thank you, Shulakkhana. আমাকে দেখা যাচ্ছে না? দেখা যাচ্ছে?
এখন ঠিক আছে।
আচ্ছা, good morning everybody. We have assembled here today to participate in a webinar on health awareness being organized by Bethune College, Kolkata, as a part of the 7-day Students Week program initiated by the Department of Higher Education, Government of West Bengal. And first of all, I on behalf of our principal, Professor Krishna Rai, would like to welcome you all. It's very sad that Professor Krishna Roy is laid down with COVID, and probably she has not been able to join. And I spoke to her family just in the morning. She's not feeling very well also. So probably she won't be able to join. It's very sad because she's very enthusiastic and over energetic about all these type of webinars. And it's really very sad that we'll be missing her today. With this few words, I would like to welcome our speaker, Dr. Shomiron Panda, who is the Director General ICMR and the head ECD. A very warm and hearty welcome to you, sir. We are very, very eager to hear from you. I would also like to extend my warm welcome to all my colleagues who are the participants of today's webinar. And last of all, I would like to extend my heartiest welcome to my dear students who will, I'm sure, will benefit highly from today's program. And Dr. Panda will now speak on the awareness of present pandemic with special emphasis on Omicron. For the last 2 years, about 2 years, we have been in a very unusual situation, and we are trying to adjust ourselves, or we have already adjusted ourselves to this type of situation. And it's a terrible situation. At least I find it terrible, sitting at home, taking online classes. The situation had just become a little better, and we were thinking that the college has reopened and we will be taking offline classes. We will be interacting with our students. But again, the situation is getting from worse to worse. So with this, what is, because we have people, I was just talking to a person yesterday who was he was employed with one of the most important pharmaceutical industry of India, and he was talking about also this situation, and he was telling me that everybody is very worried that so many people are dying of COVID, but do you know how many people die every day of cancer, of flu, and other heart cardiac failure, and brain cerebral attack and all? What I told him, I will also tell you with today, that maybe in our country we have many deaths because of these type of situations where you have cardiac failure or a cerebral attack, or some other health problems or accident or whatever it is. But these 5 lakh people, nearly 5 lakh people who have died of COVID wouldn't have died if COVID wouldn't come to our country. So maybe out of these 1.5 lakh people would die their natural death or something like that, but there would be 3.5 lakh people still living, and many of us have lost our near and dear ones in this situation. So I think what we need is to become aware, so that we will be able to combat this situation better, so that we will become aware of all the things that we should do or we should not do, so that we can obviously protect our near and dear ones. That is what is most important to me. We have lost so many lives all over the world. I think about, sir will know better, around 60 lakhs people have died because of COVID, and in our country only 5 lakhs people have, nearly 5 lakh people have died of COVID. So, and so many people have contracted COVID, and so many health issues are coming up, and also not only physical problem, we are having many mental issues also because of this type of situation. We are going through lots of trauma and lots of difficulties every day. So I think awareness is very, very essential for us to combat this situation. With these few words, I would again like to welcome our esteemed speaker, Dr. Shomiron Panda, and I'm sure all of us are very eager to hear from him. And with these few words, again, I would like to extend my hearty welcome to all the participants of today's webinar. And please, I would like to again tell you all that please keep yourself muted and your videos off, so that we are able to proceed with the webinar without any interference. Thank you. And with these few words, I will again hand over the mic to Shulokhana. Shulokhana, you can now take over again. Okay, thank you.
For your nice welcome address. Now I would like to welcome Dr. Shomiron Ghosh, Department of Zoology, for introducing our speaker. Shomiron, please.
Very good morning. We have today with us Dr. Shomiron Panda, the Additional Director General of ICMR. The man, perhaps the COVID man of our country. Last 2 years, I mean, he is engaged with all the management issues. And I mean, today, I mean, we will be astonished to learn that he have more than 35 publications over the I mean scenario of the COVID. So not only the mere research, also the policies, and in every sphere of the COVID pandemic I mean management, the pivotal personality is Dr. Somiran Pandit. It is a unique opportunity for all us to have him in this morning to share his views in present, I mean, surge of the COVID patients. The today's newspaper rocked that there will be about 23% surge in positivity in the COVID test in Kolkata only. So in that context, I mean, Dr. Panda is working over full of his tenure as an, I mean, viral. I mean, he is working with the AIDS. I mean, you know that in 90s, the AIDS patients or the surveys in northeastern countries were conducted by him, and after that, he have a sincere work on the HIV and hepatitis also. And then he, I mean, when the pandemic came, I mean, he is, I mean, one of the key personality to carrying all the responsibilities for the coordinating the COVID research and strategies and management for the country. So it is a unique opportunity, certainly, to hear from him. So I welcome Dr. Panda to, I mean, here. Dr. Panda, please, for your deliberation.
All right. Good morning. And really, thanks from my heart for inviting me during this celebration by Bethune College. This is Students' Awareness and particularly Health Awareness Week, which is being celebrated. And thank you, Dr. Miran Ghosh, who actually contacted me. And then while I logged in, I had the opportunity to briefly interact with Amitadi, Shulakshmidi. So thanks, everybody, the faculty and the teachers. While I was requested to have this presentation about the pandemic, the COVID pandemic, and with a particular focus on Omicron, I was thinking, how should I speak in terms of the media to be used? And I asked the teachers and the faculty with whom I was briefly interacting, what should be my media? And I was told that I should probably mix English and Bengali because some of the students might not be able to comprehend things if I speak in Bangla or Bengali all the time. So I will try to do so, but mostly I will use English in the best interest of everybody. So without much ado, I would share my screen, and you have to just assure me if you can see my screen.
Yes, sir, it is visible.
All right, so let me go to the full screen mode. Is it fine now?
Yes, sir.
All right, thank you. Thank you so very much. While I was invited to interact, I thought that, well, let me not keep it like a typical academic presentation, but in the mode of informal interaction with the faculty of Bethune College, and nobody needs to, add any adjective or introduction for Bethune College. It goes by its name and due to its performance in terms of training, capacity building of the students and future researchers in the country, and academicians and scholars. But I thought that it would probably be appropriate because we do not have answers to all the questions that we might have in our mind around this new pandemic that we are facing, not only in India, it's pandemic by definition. So the definition of pandemic is an epidemic when it crosses the country boundary or international borders and affects many countries, it need not be, it is not a must that the particular disease or a particular disease needs to be there in every country. But when a particular disease occurs in a frequency which is greater than the expected frequency, then we call it epidemic. And when an epidemic crosses country borders and international boundaries and are reported from multiple countries, we call it pandemic. So I thought that I will keep it open in terms of a few thoughts around COVID, including Omicron, and that's how I named this presentation, pandemic, a few thoughts around COVID, including Omicron. Now, this is a thinking tool, and I think it would help the students who have joined today morning webinar through virtual link. Anytime we think about a disease, it helps us to think in terms of host, which in this particular case is us, human beings. The agent meaning which is causing the disease. In this particular case, we are talking about a virus named SARS-CoV-2 and the environment which facilitates the spread of it. But I would draw attention of the audience here that it need not be seen as only one agent. Like if you have a look at it, the agent virus also might be facilitated by the environmental pollution. So the agent in itself might not be as effective as it would be while there are environmental pollution or crowding. And when we talk about the host, it's not just the host genetics, but it's the host behavior as well. This morning, while while I was being introduced, I was told that COVID is a reality. COVID is the name of the disease, and the causative agent here is a virus called SARS-CoV-2. It's a reality, and one of the faculties in Bethune College is down with COVID now. And COVID, the D stands for disease, and CoV stands for the coronavirus. So VI is virus, CO stands for corona, and D is disease. And it is caused by the virus named SARS-CoV-2. And coronaviruses are actually more than, there are more than 400 to 500, but those which are relevant to the immediate animal kingdom, which lives in close proximity to us, is around 40 to 50. So it's not a virus that we didn't know about, but this particular variety, SARS-CoV-2, is only one, which is causing the disease COVID-19. This is a typical diagram which speaks about the virus getting attached to the receptor, which is down below. The scale-like thing, that's a cell wall, and there is a receptor, and the virus gets attached. And as it looks like a crown, so that's why corona, the name comes from. And this is just to say that how does the mutation happen? D. When an amino acid D is I need not to elaborate this here, which is aspartate, gets replaced by G, which is glycine, we call a mutation has happened. And when we will come to the whole issue of Omicron, we will use this term mutation. So that's why I thought it would be important for us to have this sketch here. And RNA viruses in particular, this is an RNA virus we are talking about. And RNA viruses in particular, and those which do not have a proofreading system, efficient one, then you get to see many errors happening. Some of the errors, and these are called mutations, the changed situations, and some of the errors even do not turn out to be advantageous for the virus. So it is not that whenever there is a mutation, there will be more pathogenicity, etc. I will bring this issue up again at the end of my presentation. Now, when we talk about COVID-19, because it was in 2019, December, when it came to the notice of the world, so the responses does not require only doctors. It requires doctors, scientists, healthcare professionals from different walks, and also the healthcare system. So that's why, if you remember these 7 bullet points that would be helpful. This is an emergency situation. So how does governance operate in health emergency is important. And there will be necessity of healthcare in large number. So is the country prepared or was the country prepared? Will be a question. And if not, then the infrastructure development, that's another area of response, which is very, very important. How quickly do we get to know about the entry of this virus and then health system resilience? How much this can be tackled, and public health and social measures, that is called PHSM, and then drugs and treatments and vaccines, those are other issues. And then not only communication, the CIC is very important. Communication, infodemics, and the community engagement. This is something where it is not only India, many countries have struggled. And it is the communication. How do we communicate? And infodemics, everybody comes up with an information, and people do press the button of forward without necessarily checking the authenticity of an information. And that actually adds to the myths and misconceptions. So it is not only the epidemic of the viral spread or the virus causing disease that takes place, epidemic of misinformation also takes place. So in a pandemic situation, we cannot afford to therefore focus only on the virus or the disease. The myths and misconceptions around the virus, what to do, what not to do, because particularly when a disease is new, not that we have answers to all the questions. So the CIC, communication, infodemic management, and community engagement is so very important and the most challenging. I will elaborate why do I think it is most challenging a little later. So all of you are aware of it that in December 2019, I was talking about why COVID-19 that was reported or detected. Now, here again, the whole politics around COVID-19 and SARS-CoV-2 need to be considered. The detection in Wuhan, in the Guangdong province of China, does not mean that the virus was for the first time making its appearance there. It was detected there, fine, but we do not really know how did it come into existence. Was there an animal presence and then it jumped the species barrier and it was infecting humans? But naturally, we get to hear a whole lot of conjectures that, oh, China did something and there was genetic engineering. We really don't know. But of course, this maneuvering with the organisms, that has been the case for not only viruses, but bacteria. But for COVID-19 or SARS-CoV-2, we do not know for sure. Now, this particular student who was from Kerala, was studying in China, was coming back to Kerala, and he actually traveled to many cities. And why these travel-related issues are important, or mass gathering-related issues are important, we need to appreciate that, because we are talking about a virus which gets spread through infected water droplets while we talk or sing or, talk loudly or sneeze or cough. During that time, during these acts, the infected water droplets from an infected individual spreads in the surrounding or in the environment. So if there is population density, there is crowding around this infected person. So it helps us to think in terms of an infected individual surrounded by a few susceptible individuals. So those of the students who have joined today, if you are interested, you can think of the model which talks about SIR, susceptible, infected, and recovered. So that model actually is used in infectious disease related projection. So we needed to know the epidemic which was reported and the potential responses, what we could think of. ICMR, which is Indian Council of Medical Research, played its role. But then research is not the only thing that could handle an epidemic. So that's why I'm saying here, the title of it is Big Trees, Small Trees versus Forest. So whoever does a particular thing, be it a technocrat or a bureaucrat or this, today we are trying to spread awareness. There are researchers, there are doctors, nurses, frontline workers, even a Safai Karmachari or an ambulance driver. Everybody is part of the response that gets mounted by a particular country. So what I'm trying to say, that when we talk about responses to a pandemic, we need not to think that it's any one agency's or one institution's job. It requires multi-sectoral, multi-ministerial, and involvement of multiple quarters of a society and many individuals. It's not one person's job or one agency's job. Now, this particular graph, the blue line is showing the positivity. So if the testing does not happen, we really do not know, because at the beginning, while we did not start this session, in Calcutta, yesterday it was 17% test positivity rate. I got to see this on TV, and now it is around 20 or 22%. That means the infection is spreading. But not that all the infections which are happening in Calcutta or in Delhi or in Mumbai are Omicron. Delta is still the predominant variant. When we say variant or mutant is the same thing, and mutations are expected. Mutations do happen more so for RNA viruses. I am repeating myself, as I said, that if RNA virus cannot rectify the errors remain during the proliferation, the error in terms of one amino acid getting replaced by another. Sometimes part of a gene gets deleted. Those kind of major changes are called shifts. But when the changes are minor, like one amino acid gets replaced by another, we call them drift. These drifts and shifts. The drifts are smaller changes and the shifts are bigger changes. So those were known even during the pandemic, influenza pandemic, or flu pandemic, which wrongly is called Spanish flu, because Spain actually reported very efficiently. But that does not mean it got originated from Spain. Actually, there were many infections happening during 1918, the Spanish flu year, as it is wrongly known, because it was actually a pandemic, so it was not restricted in Spain, Mumbai, and in Calcutta. But there are very few records, even though those times, from within India. If you are interested, I can tell you about a book which has some description of the pandemic happening in India and hitting India. But that was 100 years ago. That was over the flu pandemic. But if coming back to this particular slide, the green bars talk about the tests. So if you do not have the test mechanism while a new virus hits the country, then you won't be able to really know how quickly it is infecting people or what is the positivity rate. So in India, we had this laboratory, BSL. It stands for Biosafety Level 4. So different organisms can be handled with different levels of safety and security arrangements. So there are laboratories BSL2, BSL3, BSL4 is the highest level of containment, Biosafety Level 4. And there is only one in India, which started operating in 2012. That's located in Pune and at the National Institute of Virology in Pune. And Wuhan lab, and the Wuhan, which is a known city name to everybody now, they had their BSL-4 lab established and functioning in 2017. Why am I making this point or underlying this point is for the same single important reason that India was prepared, and this is ICMR Institute at Pune. There are also another institute called National AIDS Research Institute in Pune. So NIV had this BSL-4, and that's why we were ready. So we must give credit to the predecessors. So nothing gets done overnight. So preparation and farsightedness becomes very much required. if a country needs to remain prepared for upcoming pandemic and epidemics or outbreaking disease. Now, while COVID hit us, you see on the left-hand side of this blue arrow, which is down there horizontally, only the travelers were tested because there was report that cases are happening in Italy, UK, China, many other countries with travelers were tested. And then gradually the healthcare workers and SARI stands for severe acute respiratory infection patients and their contacts, they were getting tested. So gradually more and more population sectors were included. And now we have attained a stage where anybody requesting a test can be accommodated and can be tested. Now, about the data. Now, I did talk about the data and then treatment and then vaccines and the drugs. So anybody and everybody can generate data. Why am I talking about the need for having important or good quality data? Because data everywhere, but not much data to use is the situation which happens if we do not become careful at the time of designing a survey, a surveillance, or even an experiment. So this COVID Kavach ELISA is an ELISA test which detects the antibody that gets developed following exposure to the virus. Now, this was developed by the NIV, the National Institute of Virology, located in Pune. That's an ICMR institute. And due to this indigenous development of the ELISA kit, we were able to do the national or conduct the national sero survey. Sero survey is known as serological survey. The name comes from the fact that we collect serum, and in the serum we detect antibody. And when there is antibody, it says that, well, this particular individual got exposed or infected with COVID, SARS-CoV-2 virus. The name of the disease is COVID. So SARS-CoV-2 virus, that's why now we are detecting antibody. And that actually allows us to know how quickly the infection is spreading and what proportion of people in the country have become infected. So now you see May, June 2020, we had very little, 0.7% of the people were infected. When in August and September, it was around 7%. And then in December, January 2021, this is after the first wave. The first wave peak happened in September 2020. After that, almost 16%. And then June, July 2021, there was another national serological survey which proved that 60 to 70% of the people are already infected. So you see how the proportion of people in the country who were exposed, and some of them were asymptomatic, but they were infected, and some developed symptoms. But the proportion sort of reached a height of 70 to 80%. And then if you take vaccines, you realize that antigenic exposure to the people are not only through vaccines, but also through natural infection. And many infections, 70 to 80% remain asymptomatic. Some of the individuals get to symptomatic stage of infection. So when you are taking vaccine, after vaccine, if you get infected, or even prior to getting vaccine, if we get infected asymptomatically or symptomatically, it's actually twice we are getting exposed. So if you have got 2 doses of the vaccine and you have been infected, you have actually received 3 exposures to the antigen. So your immunity will be getting charged accordingly. Now you can see that while doing the test, we take, and while I am talking about the test, RT-PCR or rapid antigen test or home-based test, we do use a nasal or nasopharyngeal swab, which is pushed through the nasal cavity. And there is again oral, to the oral cavity, oropharyngeal swab is taken. India did not even have the swab in sufficient numbers, but now we need to not only produce swabs, but also export them. Similarly, RNA extraction kit. I was repeatedly talking about the SARS-CoV-2 virus being an RNA virus. So what is done? I'm talking to the students mostly. The faculty will not be told about this, but I think it would be quite interesting for us to know. After taking the swabs, these are kept in the test tubes, and the test tubes have the viral transport media, or VTM, as you can see on this particular slide. So this VTM tube is then capped, and the tube is transported to the laboratory. In the laboratory, the RNA extraction takes place, and then RT-PCR test, which is polymerase chain reaction, that takes place in order to figure out whether the virus is present in the clinical specimen, which was collected through the swab, nasopharyngeal and oropharyngeal swab, kept in the viral transport medium and transported to the laboratory. That is what is examined through the RT-PCR test. Now, there were inquisitive minds, who thought, can there be a method where we can have RNA extraction free method? Like, we need not to put it, we just put the swabs in the test tube, no VTM, and you send the test tube, you use elute buffer in the laboratory, and then from that elute, after a little bit of heating, can you do the RT-PCR test? So if you are able to do that, then you are saving money for VTM. Even if the test tube accidentally breaks, then you do not have the spillage, and then RNA extraction reagents and the instruments and the trained human resource, all those can be saved. Time can be shortened, money can be saved. So there was this, there were many protocols which thought of RNA extraction method. And then the students who are today, I mean, they are the future researchers or workers. So they have to ask this question: does this method work? If it works, what is the sensitivity and what is the specificity of this method compared to the old gold standard method, like the VTM-based transportation followed by RNA extraction followed by RT-PCR. So I was fascinated by this book. I thought that I will share this cover page of this book. It has got nothing to do with COVID-19, but I think that the students who have joined through the online platform, the virtual platform today, in relation to this health awareness week celebration, they might be interested to purchase this book and learn from here that how one need to remain aware of different nuances around experimental design. So I thought that I will just keep it like informal sharing about the books that I thought was interesting. I keep on buying books, not that I'm able to read all of them, but I always get attracted towards books which stimulates some kind of interest in me. In terms of treatment, when we talk about treatment, these are the symptoms, but not that the way Omicron is spreading so fast, the home-based care would be the key, because home-based care can handle the situation. If the oxygen saturation is staying above 92%, you should not worry. Everybody should have a pulse oximeter at home, and that's possible, quite possible. It does not require any sophisticated technical ability to measure oxygen concentration. So pulse oximeter and thermometer, these two things should be quintessential elements for home-based care. We can measure our temperature, and we can also measure oxygen saturation. Although I thought that I would speak for 30 minutes initially, but I might go to 40, 45 minutes, and I hope that it's going to be okay. Now, given the, coming back to the topic, the Omicron is spreading fast. It is not possible, not even required for everybody to go to the institution. It is possible to detect the infection, possible infection, quite fast. So if there is headache and fever and a little bit of sore throat, then we can do a self-test. So these are rapid antigen tests. These are not RT-PCR, but if I'm symptomatic and if rapid antigen test or RAT in abbreviated form, RAT, comes out to be positive, it has high sensitivity. The rapid antigen test has a high sensitivity for somebody who is symptomatic. So that should be okay. And then I can isolate myself at home, and family members or those who stay at the same home, they can also have the COVID appropriate behavior. From ICMR, we created this home-based care one-pager, and it talks about how to take good care of ourselves. It is going to change as we now know that some of the medicines which are thought to be useful do not really work well. And we need not to unnecessarily take antibiotics. We should always have a consultation with our treating physician. The National Task Force looks into clinical research. The diagnostic, I was talking about the RNA extraction-free method to detect SARS-CoV-2 virus. Epidemiology and surveillance. Epidemiology is a very interesting word. Epi means upon, demos means people, and logos means knowledge. So it is knowledge upon people. It could be related to some health practices. It need not be always related to disease. But of course, in disease, you can have surveillance and you can have epidemiological investigation. But any behavior or practices which is happening in large numbers, for example, mask use behavior is also an epidemiologic topic which can be investigated, should look into. When are people interested in using masks? So I will draw now your attention to the triangle. This triangle is called epidemiologic triangle, host, agent, and environment. So the host behavior is also a topic of the epidemiology. So it need not be, as I am repeatedly underlining, it's not about the virus only. It's not about the disease and the treatment. It's about human behavior. It's about the ability of a susceptible individual to come to close proximity to an infected individual. Because if you go to New Market in Kolkata or Sarojini Nagar Market in Delhi or in a suburban place or train in Mumbai, we can see how is the behavior, happening in the mass. And no point blaming, but how interestingly we can engage the community. That's why I said that CIC is the most challenging area. The last C is community engagement. So engaging the community, even the students at Bethune College, how they can be engaged in innovative behavior change communication, not only in the college, but also in their respective localities, is a very, very interesting area. And that actually falls under operation research. Another example of operation research is delivering vaccines in hard-to-reach areas or to hard-to-reach population through drone. So drone-based vaccine delivery. Now we have, gone through several iterations and the guidelines get changed. Vaccine development, all of you know. This is a website which one can always log in. This is the ICMR website, and you will get to learn about the vaccine development initiatives for SARS-CoV-2 and also against many other diseases. So this is just for the interest of the students. If any one of you is interested, please feel free to get to the ICMR website and then to the vaccine development related portal. Now, on the left-hand side, this is an electron microscope picture of the coronavirus. You can see the roundish structure, and then there are crown-like things. The sketch that we saw on one of the slides, this is actually a photograph taken by the scientists working at the National Institute of Virology, and that's how it looks like under the electron microscope. And then on the right-hand side, you are seeing the upper panel, the reddish color. That's a cell line which is not infected and affected by the virus. But when you do the virus culture, which is vero cell line, the green one, it's immunofluorescent picture. And you see the cells are confluent. This is called syncytia formation. So you get to see the damaging effect of the virus. The bullet point below is very important. India became the 5th country globally to isolate the virus and culture it. Because once we culture the virus, then it helps you to develop the COVID Kavach ELISA that I talked about. So it actually helped in getting the tests done. So that's why it was such an important step. Now, when the virus came in, whether the phase 2 trial results can be used, there was a whole lot of debate. The phase 2 trial is not completed. How can we, have the virus receiving emergency approval? But that's wrong. If there is a disease which is life-threatening, which causes serious disease conditions and leave us with disability, and this is called unmet medical needs. And under a condition of unmet medical needs, it is allowed to use the phase 2 clinical trial results. You need not to wait till phase 3. So that's absolutely fine. And in clinical trials, like whenever there is a medicine or whenever there is a vaccine, what is attempted to find out is the risks. Is that how much is the risk associated with the vaccine and the benefits, and the risk-benefit ratio actually guides us. And then slowly it is rolled out, and there again community engagement remains so very important, because people might be unnecessarily afraid of a new medicine or a new vaccine that is rolled out. Please, please, please remember that the vaccine does not prevent infection, but vaccination does, and that's true for other infections. But in SARS-CoV-2, so here I'm highlighting that only having a vaccine is not good enough. There always remains vaccine hesitancy. And particularly in SARS-CoV-2, we know that the vaccines modify the disease. So even if I'm vaccinated, it is important for me to use masks, because these vaccines will prevent me following infection to go to the severe form of the disease, or to get admitted to the hospital, or the situation where I might require oxygen, or there could be unfortunate event of death. So the vaccines against COVID-19 or SARS-CoV-2 actually are disease-modifying vaccines. So I would say that there is no need for vaccine hesitancy. We should go ahead and get the vaccines. But following vaccination, we should also use masks, because these vaccines do not prevent acquisition of infection. So if I get infected and if I'm vaccinated, maybe I will not go to the advanced stage of the disease, but from me, others can get infected. So as a responsible citizen, it would be important for me to use masks, even if I get vaccinated. India had a huge experience of immunization program. This is about children. But right now we are talking about the disease which is about adults and children. Actually, children handle the infection much better compared to adults. The adults who have diabetes, hypertension, or other comorbidities, they get to the advanced stage of the disease. So adults also have vaccine hesitancy much more, because for children, the parents take the decision. So what I'm saying, that India had a huge experience of dealing with the vaccination, and that's why it had a robust system, but there were additional challenges as well in terms of COVID-19 and the SARS-CoV-2 vaccine. Now, communication, CIC, the thing that I'm repeatedly talking about, communication, infodemic management, and community management or engagement. So you see, this is from 100 years ago. And people were inviting others to attend a meeting, and the meeting is about anti-mask. And tonight at Saturday, you are invited, January 25th, and Dreamland Ring, and there people are being, asked to gather to discuss why should we not use masks. And on the right-hand side, if you see, there are different style of using masks, different types of masks. So it's not today we see people using masks around the chin or the neck. It was there even 100 years ago, you see, and somebody is even smoking a pipe through a hole made in the mask. So this is all about behavior. So coming back to this whole triangle of agent, host, and environment. It is so very important to address this issue. If you are interested to read things on face mask, you can type face mask IJMR, Indian Journal of Medical Research, and you will get some interesting write-up there. IJMR, Indian Journal of Medical Research, is a freely downloadable, journal issue, and I request all of you to freely access them. And there are COVID special issues if you are interested. Now, this is just a few examples of the cover pages of the IGMR that I was talking about. On the right-hand side, the fifth issue has come out, and there will be the sixth and the last COVID-19 special issue. But all of these are freely available and downloadable. Now, why I'm showing you the journal? The journal is one way of communicating, because this particular slide was highlighting upon the communication. And the communication need not be through newspaper or through social media, but also through the journal issues and advisories on the ICMR websites or press briefing. So we have to really manage the infodemic well. Otherwise, myths and misconceptions will spread all around. Because we also got to hear that if you take papaya leaves, the papaya tree, the leaf, the green one, you boil them and then drink the solution, and it will prevent you, or it will be a great benefit for increasing the platelet count if somebody is infected with dengue. These have not been proven to any scientific experiments. So that's why I showed that blue book, which probably students will feel like buying and understanding what are the nuances that we need to think of. And if we really want to critically ask any question, so anything can be asked. That's my point. Not to believe anything and everything which is being floated in the social media. Of course, while there was national lockdown, this is an example of the Ministry of Railways, Ministry of Law and Justice, Niti Aayog, Ministry of Civil Aviation working together. Because then the ELISA kits or the test kits were being transported at different corners or to the different corners of the country. So you see, the pandemic response requires not only medical fraternity or bureaucrats, it requires interministerial collaboration, which is so very important. 1947, we were receiving, the lady here is Maharani Amrita Kaur, who actually donated the land and building for establishment of All India Institute of Medical Sciences in New Delhi. And she is receiving the vaccines of polio and other vaccines for children from Canada. In 2021, India was giving COVID-19 vaccine to Canada. So that's the phase we have passed through or crossed through. So it's quite a bit of development. That is not to say that we have achieved everything in every field, but this is really very interesting and important to know that 1947, we were receiving vaccines donated from Canada to India. And in 2021, we did give vaccines to Canada. So that underlines the immense height that India has reached. And that's why I am showing this to the students and the young minds today who have connected to this virtual platform. This talks about the guideline, which is freely available, how the vaccination was rolled out. And there was actually a very good NECVAC. I'm the member of the NECVAC as well, the National Expert Group on Vaccine Administration for COVID-19. And it had the block level structures, the district level structures, and the state level structures. So it's not easy. It's easy to say that vaccination, well, it should happen this way, that way, but it requires huge planning and infrastructure development. Now, coming to the Omicron, I'm gradually coming to the last part of my deliberation. Useful facts are that the vaccines, which I said, the COVID-19 vaccines are disease modifying. And the full vaccination, meaning 2 doses of the vaccines, are more than 90% effective in protecting against severe illness and hospitalization. And that holds true for the Delta variant as well. An acquisition of infection post-vaccination is a possibility. You can acquire infection, you can be infected. So that's why you need to use masks after vaccination. The point I'm trying to make here is that it is not Omicron everywhere in India now. Yes, Omicron is spreading, but in some of the states, the upsurge that we are seeing, the predominantly circulating strain is Delta. So we need not to think that Omicron is spreading, Omicron is going to cause mild disease, so we need not to care. That will be a wrong perception. So it is Omicron at some places, it is Delta at some places, and in some of the states, we are seeing both Omicron spreading and also Delta spreading. So the population density of the mass gathering, which creates an environment for spread of the virus, will facilitate the spread of different kind of variants. So it is very, very important for us to observe COVID-appropriate behavior, to avoid mass gathering, to get ourselves vaccinated, and not to have travel, which is non-essential. Like we might think, oh, okay, everything is over, so let's go to Sri Lanka. So let's go to somewhere else. And you go to Sri Lanka, you get infected and you get quarantined there, and that's no fun. Now, the Omicron is a variant of concern, or VOC. As you can see on the left-hand side, O indicates Omicron, but there are different terms. variant of concern, which is actually a mutant or a mutation. We are talking about a mutant of interest or a variant of interest or variant of high concern. So these terminologies are ascribed, and it is not true that if a particular variant is called variant of concern, it is going to remain like that forever. No, they also change the position. So the concern comes from not only the number of mutations. Omicron, as you can see below on the slide, in the middle, I'm talking about Omicron here. It's heavily mutated, 50 mutations overall, and 32 is the spike protein. The spike protein is the picture which I showed. The spike protein gets attached to the host cell receptor. But then, remember, please remember this, that if a virus needs to spread, the virus cannot work on its own. So it is dependent on the host behavior. The coughing, sneezing, talking, singing, going to a crowd. So an infected person doing all of these and getting surrounded by susceptible individuals will spread the infection. But if a virus which kills its host, a dead man does not cough, a dead man doesn't talk, a dead man doesn't sing, a dead man doesn't sneeze. So the increasing transmissibility does not necessarily go hand in hand with fatality. Because if the host is dead, then the virus cannot spread anymore. So Omicron might be highly transmissible, but it does not necessarily mean that it is going to be fatal as well. But we do not know how Omicron is going to behave in elderly or in those individuals who have some form of comorbidity like diabetes, cancer, hypertension. So it is very important for us to realize that we cannot afford to infect elderly or those with comorbidity, because we have to live like a responsible citizen, number one. And number two, it is not only Omicron which is circulating in India. In India, the predominantly circulating strain is still Delta, and in parts we are seeing Omicron, and in parts it's Delta, in some of the states it's both. So it's very, very important for us to avoid mass gathering, to avoid non-essential travel, and also to adhere to the mask usage. You see, now, formerly VOI on the right-hand side, extreme right-hand column. So what I'm saying, on the extreme left-hand column, these are the code of different variants, and on the extreme right-hand side, formerly VOI, meaning formerly it was variant of interest. So variant of concern, variant of interest, variant under investigation, all these are the terminologies which are not carved in stone. They do get changed. But previously, people used to talk about UK variant, India variant, South African variant. That's the wrong way of naming a variant, because it adds to the stigma to the country, which is no good. So as concluding thought, we are all talking about third dose. The third dose might be necessary or might not be necessary, depending on the country situation and whether the person got infected or exposed to the virus and then received vaccines. That means he or she has already received the vaccine. So those are certain considerations. So let us get guided by science. This is a graph which is from South Africa. The blue line talks about the increasing number, and the red line is about death. You see, for the first 3 waves, which was like, during March and April in 2020, and then during December 2020, and then July 2021, the death was very closely following the infection. But this December 2021, the infections are going up, but the deaths are not happening in that number, which is an indication that Omicron does cause milder form of the disease. And we see here the upsurge of infections, but not necessarily the upsurge in severe cases, or upsurge in hospitalization, or upsurge in oxygen requirement, or upsurge in ventilator use, or upsurge in the number of deaths. However, let us all remember that the upsurge of infections that is being observed in India is not just by Omicron. I'm repeatedly saying that it's a mixed picture now in India. So we have to be very careful. And we cannot afford to lower our guards down. This is a very nice picture where you can see the blue is one variant, the light blue, and then the deep blue, and then the orange color, and then the red, extreme right. Red is Delta, and Delta has replaced, Delta is the predominant strain, and the earlier circulating mutants got replaced. But it could be that Omicron would replace it, but Omicron has not replaced it as yet in India. So we need to remain very careful in terms of our behavior, in terms of our hesitancy around the vaccine. These three books are very important. I keep on buying books. People take books from me within my family and outside my family and never return them, which is a bad practice, I guess. But I hope that if they have not returned the books, they should read them, which they do or don't, I have no clue. But the students might be interested in, going through these three books I'm showing here. The middle one is about the smallpox eradication. That was a pandemic also. On the left-hand side is about the pandemic century, fantastic book. It has talked about different pandemics. And on the right-hand side, those of you who are inclined to understand the disease transmission dynamics and the projection, this is a very, very good book, very well written, The Rules of Contagion. You can buy them. You can get married to the books. I think that's much interesting, much more interesting. Sorry for this comment. However, I sincerely feel that books always give you something or other, even if it is an old book that you are holding onto yourself and carrying it yourself. So if any of the students buy any one of these, they would see the beauty and the fun of it. And I think that SIR model that I was talking about, susceptible, then infected, and then recovered. All these can be understood very well from the book, which I'm showing by Adam, written by Adam on the right-hand side. The smallpox eradication story is fascinating. And on the left-hand side, you will get a fantastic glimpse of how the epidemic spread, how the pandemic happened. Pandemic, again, I will repeat, when the epidemic is a disease occurrence, I will give an example also at the end. Disease occurrence much more than the expected frequency. And when the epidemic cross the country boundaries or international boundaries, then we call them pandemic. It is not a must that every country needs to be affected, but many countries getting affected is good enough to be called it a pandemic. This is the recent phenomenon that is being observed in Delhi. This is an upsurge from 14th December to 27th of December 2021. And clearly, there's an upsurge. Not that all of these are due to Omicron. Many of them, half actually, are due to Delta as well. So if the mass gathering is happening, if we are lowering our guards down, it is possible for us to get infected. Those who are vaccinated might face less severe form of the disease, but those who have partial vaccination, meaning first dose only, or those who have hesitated and have not received the vaccine, they have a chance of getting to the severe form of the disease. That's true not only for Delhi, it's true for Calcutta as well, or for any metropolitan city, or even in rural areas where people congregate for political reasons or religious reasons or social reasons, like a large marriage party. So it's all about the population density, a conducive environment where the virus can spread from an infected individual to susceptible minds around that infected individual. Now, I will request the students to have a look at it. These are studies from Japan, UK, and Hong Kong, and they showed this is about Omicron, and the lung cell infectivity has been found to be less in all three studies. These are in vitro laboratory studies with hamsters or small animals, and that's why we know that, and there is data also to indicate that Omicron is causing less severe clinical disease, but Omicron is not the only variant which is circulating in India. So I will stop here, and I will request students to look into what is written here. The best match, meaning matchstick in the world will not light a candle unless the wick be first suitably prepared. दिया जलाना होता है तो वो जो विक होता है जिसको हम कहते हैं बाती जलाने के लिए वो प्रिपेयर करना जरूरी है. So I did talk about the DSL-4 laboratory. It was there prepared in 2012. There were scientists prepared. There were social workers ready. There were, the students from zoology, life science, botany, molecular biology, and they were all there. That's why we could quickly assemble the team, the country, across the country, and could respond. Thank you. I will stop there.
Thank you, sir. Thank you very much for your very informative and vivid lecture. Now the floor is open for question. And already we have got 2 questions in the chat box. May I read it, sir, for you?
Sure.
Okay, the first question is asked by Dr. Gautam Goswami of Physics Department. He is asking that it is often discussed that Omicron is many times powerful as a spreader compared to the previous variants. What is the measuring stick to determine the spreading? Is it the virus or RNA density in the ejected droplets from the infected person?
Okay, so the transmissibility of the virus is in the population setting measured by the doubling time. So if today in a specific geographical location and within a population group, 10 people have been found to be infected with Omicron, and after 3 days, it becomes 20 or 25, the doubling time is 3 days. Whereas for Delta, earlier, it was found to be 10 days. So that is the way transmissibility is measured. So when the doubling time of a particular variant is measured and it is significantly lesser compared to the earlier circulating variant, we say that this is highly transmissible. And of course, the changes in the viral variant works for this or adds to this transmission efficiency, but it has been found that the same structural changes were not able to infect the lung cells. So transmissibility was an advantage that this particular variant gained, but the same variant would not become fatal to the lung cells, and that's why the clinical disease was happening in a much less severe form. As the question has been asked by a faculty from physics department, I must also say that the SIR model that I was talking about and the reproductive number that I'm sort of alluding to now, many physicists from within India have worked on it, and we have also worked on the projection. So many people talk about prediction, but looking into the transmissibility, the vulnerability around in the society, the vaccination rate, the projections are done. But I thank you, first of all, for this question. It is very important. That gave me an opportunity to discuss about the doubling rate.
Okay, thank you, sir. The next question, I hope that she has already got his answer, that it is more harmful than Delta variant. You have already answered, sir. The next question is been asked by Snigdha Sri Manna. She's asking that what's the difference between variant of concern and variant of interest?
All right, no, that's a very good question, Snigdha. So if an individual is identified to be infected with SARS-CoV-2, so he or she is an infected individual. So you get a nasopharyngeal and oropharyngeal swab, you carry it in the viral transmit media, the RNA extraction takes place, and then the RT-PCR test is done. But when the positive test result comes out, you can use that RNA material that you have with you for doing the sequencing. And while you do the sequence, you see that, well, the structure of the virus is such that it is differing a bit from the earlier circulating strain or the wild strain. So you say, oh, okay, there is a change we have identified. So it's a variant of interest. But then whether it is variant of concern or not, that is dependent on 2 or 3 things. First of all, if in the laboratory this particular newly identified variant causes the cell-cell fusion, or if there is a very quick cluster formation in the population level, or if the people are getting into severe form of the clinical disease, or deaths are happening. All these 4 or 5 factors, maybe one of them would immediately tell, oh, this variant is a variant of concern. If the structural changes, so many mutations, like Omicron, and it is not only 32 or 50 mutations in the Omicron, in the viral genome, but also its ability to transmit so rapidly makes it a variant of concern. So variant of interest or variant under investigation, VUI, is also another term which is used. So VUI or VOI are the terms when a particular variant which is identified following sequencing is being studied. So it is under study, but there is no concern, immediate concern from public health perspective. But when there is a public health perspective, there is immediate concern of it spreading fast or killing people or causing, advanced clinical form of disease. Then the term variant of concern.
Okay, thank you, sir. Dr. Shatarupa Bandyopadhyay is asking the question that, is COVID-19 a genetically engineered virus? If not, and if it is a natural virus, why hadn't it spread before?
A very good question. The answer is, I don't know. Neither does anybody know in the world, or we cannot come up with a firm answer. Yes, the virus, the coronavirus were there in the nature, infecting animals. Maybe there were mutations which imparted a special ability to the virus to jump the species barrier. Maybe somebody did some more genetic engineering also on the coronavirus. So in essence, what I'm saying, coronaviruses were present and are present in the nature, infecting animals. Not that all of them can cross the species barrier, or jump, as we call it, jump the species barrier and infect humans. But mutations, sometimes it is known to impart that ability. But whether that has happened with SARS-CoV-2 or there was a genetic engineering as well, we do not know. But time will say, time will tell, and the investigations are ongoing. Right now, no definitive answer can be given, but these are the possibilities, of course.
Okay, sir. Devoshmita Ghosh is asking, can masks prevent the transmission of COVID-19?
Well, if we say COVID-19 as a disease and SARS-CoV-2 as a virus, yes, use of mask, but not a chin mask or a neck mask. It's a mask which covers the nostrils, the nose, and the mouth properly. And using a mask does not even say that, well, you are using a mask so you can go to the crowd. No. So it is multiple behavioral factors. So it is mask use along with other COVID appropriate behaviors like the hand hygiene and avoidance of mass gathering, be it for the political reason, social reason, or religious reason, would help us preventing further transmission of SARS-CoV-2 virus.
Okay, sir. Now refer to your concluding thoughts in the presentation, the slide that was showing that the third dose decision has to be guided by side effect profile. So Shampriti Baul is asking, she is actually asking for some clarification or explaining this statement. What does it mean by the third dose decision has to be guided by side effect profile?
Sir, you have been muted.
Okay, so sorry about that. Basically, when a third dose will be considered, should it be the same vaccine which was used for the first dose and the second dose, or it should be a different vaccine? Now, this approach of using a different vaccine for boosting is called heterologous approach. And if we use the same vaccine, we call it homologous approach. Now, the vaccine which was used for the first dose and the second dose, they have come into the program because they went through the safety profile and the risk benefit analysis that I was talking about. So if you use the third dose, that's it. There is no worry about it. But we really don't know whether using the same vaccine would have the desired boosting effect. So now the data or the evidence is being generated from the country, throughout the country, that a different vaccine used as a third dose has a better boosting effect. But there again, one has to see that as it is a different vaccine which is being used for boosting the effect, the immunity, is it safe or it might give rise to some side effects? As of now, there is a very good Lancet paper, and the safety and the reactogenicity study is indicating that side effect or safety profile is fine. The heterologous approach is fine, provided those vaccines which have been tested for the boosting are used. So that's why I was just bringing this issue up in terms of people appreciating it is not the homologous approach which might be used, it could be heterologous approach, and there the safety concern need not be bothering one if there is scientific evidence available.
Okay, thank you, sir. One more question in the chat box. May I ask it, sir?
Sure.
Yes. Those of us who already have been inoculated with both the doses of COVID vaccination and had prior infections of COVID-19 as well, are we safe? Can Omicron dodge the antibodies we have?
All right. So yes, I can tell you that if you got infected and also received your vaccine, as I indicated, you have been exposed to antigenic challenge. Your immune system or your body has been exposed to antigenic challenges or exposures. So I'm sure that your immunity has played its role. You need not to worry. Just wait for this third dose related decision. But safe in terms of acquisition of infection, no. Meaning it is quite possible for us to acquire the infection. Maybe we will not get into the severe form of the disease. But as I said, these vaccines do not prevent acquisition of infection. They prevent us to go to the severe form of the disease if you get infected. So I will say, yes, you are safe in terms of not getting to the severe form of the disease, or not requiring hospital admission, or not requiring oxygen administration, or not requiring ventilation if you get infected. But from the point of view of acquiring infection, you and I and any one of us is quite capable of acquiring the infection, passing it on. So acquiring the infection and passing it on to others won't be a very good idea. So I would say that yes, you're fine. You need not be worried. You will be okay if you get the infection. You have very, very, very little chance of getting to the advanced stage of the disease. But from acquisition point of view, you are capable of getting infected and passing it on to others.
Okay. Thank you, sir. Thank you for explaining all our questions with such patience. There is one more question. May I ask host, how long will we continue the program? That means questions are actually flooding in the chat box. Two more questions now.
Perhaps that would be the. I mean, these two will be the last because it's already 12:30. I mean, Sarah will also previous engagement also.
Right, sir. Okay. Shomiranda, I'm asking the last question. As we know, the situation of coronavirus and Omicron remaining same, and it continuously affect on our health and study concentration. So, why someone can't get some strict decision about this situation?
Okay.
Okay.
I read about another children-related issues also. I will respond to both. One is that, what about mask use? Whenever we talk about mask or using mask, we talk about COVID-19 or SARS-CoV-2. I would say that's a very restricted view, or a view with blinkers. You know what I mean. Why? Because if we use masks, it is not only going to protect us against SARS-CoV-2, but tuberculosis, influenza virus, other respiratory tract infection causing viruses, and even the air pollution. So, use of mask as a social norm or our individual behavior, like we wash our hands before taking food, similarly, using masks and making it a norm would probably be very beneficial for us, because the way human beings are dealing with the environment, animal nature, we do expect more and more infections, epidemics, and pandemics happening. So in Japan, in Thailand, and in many Southeast Asian countries, mask use while going out in the public is a social norm. So if we understand this, and if we adopt this social norm, it would be quite beneficial for us as a whole. So I would insist that we do. And I am a strong believer of us, people, taking responsibility. Government has its own responsibility, but what it would do or it would not do, I cannot leave my health to government's hand, because government comes and government goes. Political parties come and political parties go. But if we are to remain concerned about our own health and about our near and dear ones, we can take certain decisions and we can make certain behavioral changes so that it becomes the healthy hygienic practices become the social norm. That's number one. The other question that I saw in the chat box is about children. The fourth round of National Serological Survey, I showed on one of my slides that trend. And the last National Serological Survey happened in July, August this year. And we realized that more than 50%, actually 60% of the children have been exposed and infected and showed the presence of antibody while they were tested with the ELISA test. So that means while the schools were closed, even then the children got infected. So where from did they get the infection? They got the infection from the adults. But while that high proportion, 60% of the children got infected, we did not see many children coming with severe form of the disease to the hospital. So that clearly tells us that children do get infected from surroundings, but they do not necessarily fall sick. Now, 15 to 18 is actually adolescence. And in that age group, adolescents do mix with adults, go out in schools and colleges. So their mixing pattern is much more in terms of the numbers and the frequency compared to the younger children. So that's why 15 to 18 age group children, whom I call adolescents, are being vaccinated because if they get infected, and many of them might be suffering also from obesity or diabetes, which is comorbid condition, and there is a great deal of risk of them getting into the advanced stage of the disease. That's why it is very important for 15 to 18 to get vaccinated.
Thank you very much, sir. We have come almost at the end of the session, today's session. Now, at last, I would like to ask a very simple question from a common man's perspective, that with the present vaccination schedule in large scale, and also with the safety measures continue to be maintained, and the virus is getting mutated so many times, so fast, how long would we exactly expect to combat the pandemic completely?
Very good question indeed. And you have highlighted the multiple mutations and that are happening in the virus. So we have to mutate in terms of our behavior. So we have to mutate in terms of the way we deal with nature. So if we also change in terms of handling the nature, being in harmony with the animal kingdom around us, and the mass behavior, certainly we'll be able to make it a better world, and we'll also be able to live healthy in the changed world that we all dream for or we are aiming for in the new year 2022.
Thank you very much, sir. And now it's time to say a final Thanksgiving. And for this, I would like to request Dr. Shatarupa Bandyopadhyay for offering the vote of thanks to conclude the program. Shatarupadi, please.
Thank you, Lakhana. It was indeed a unique talk, and throughout we all perhaps was mesmerized, and Shomiran was absolutely correct that it is indeed a unique opportunity to have sir with us for both students as well as teachers of Bethune College. So as for the formal vote of thanks, may we thank our honorable speaker today for having us with him and spend his time with us. It was an extreme privilege for all of us. Thank you, sir. And we are hoping that we will get to hear from you. And if we invite you in future, you will give time as well, just like today. And I might share a little information with you that during the second phase of the pandemic, our Bethune College students, they made a Bethune College COVID squad, and they tried to help all the teachers and students and other persons in our community to help their bit to do their bit. And after that, I thank our principal ma'am, Professor Dr. Krishna Roy, for giving us this opportunity and always guiding us to these kind of programs. I also thank Professor Kamal Kanti Shom, Dr. Kamal Kanti Shom, Coordinator IQSC, Dr. Amita Kaur, Secretary Teachers Council, for being there always whenever we are in crisis or whenever we are in need, get their guidance and advice. I also thank the teams actually behind this program, the Students Activity Cell members, NSS members of NSS unit, and Students Committee of Bethune College for organizing this program. And I also thank the teachers and student attendees from other colleges and from our college as well. And we should not forget our non-teaching staff friends also. It was a wonderful morning, and I think all of us was truly enriched with the information, with the insight given by sir. Thank you, sir. Again, over to Shlokhan.
Thank you, Shatarupadi. So I think we have finally ended our session. Thank you all for giving the interest and attention. And I would request all the participants to please fill in the feedback form if they have not done it as yet. Thank you, sir, for coming.
Thank you. Have a wonderful Sunday ahead.
Thank you, sir. Thank you.
Thank you, sir.